student and just kind of talk to everyone here? Welcome.
I can introduce the student. I don't know that I can introduce the project.
Sure. Okay. What am I?
Oh, sure, however you like.
Okay.
Ready.
All right. Well, thank y'all for being here so early in the morning. 8 o'clock. Yes, everybody likes 8 o'clock? Yay. All right, so Taylor's been working with Dr. Turner, and is going to probably tell us something about C. elegans, I'm guessing, right? And she's been working with her for about 1.5 years now, and actually it's in Gen Talk, so it's been a lot of fun to be introduced to a student that I have, so that's really great. So, Taylor, why don't you come up and tell us about your project?
So I've been doing research on congenital stationary night blindness. It's a genetic condition found in humans and horses. Basically, it inhibits your ability to see in limited or no light conditions. It's associated with the TRPMI gene, which basically, when you see in limited light conditions and your photoreceptors get those signals, when they're attempting to get it to your brain, that pathway doesn't actually function. So if you can, I don't know how to zoom in, but over here, a typical pathway has this sodium and calcium channel, but when this mutation is present, that channel is no longer there, so it inhibits that signaling. Specifically in horses, this TRPM1 mutation is found to be associated with what's called a leopard complex pattern. It's where you basically will see like white streaks on hooves or very large white patterns that occur all over a horse. So that's kind of probably more what it's known for, because it's typically associated with the night blindness. I specifically work with a C. elegans model or a nematode. They're just very small, clear roundworms that actually have a very similar genetic makeup to humans. So it makes them really nice, very small, very cheap models to be experimenting on. And those are actually, there's a picture of them right there. So my hypothesis is that if this specific mutation in the nematode ortholog that I'm dealing with, which is GON2, it should, if this mutation is induced, it should cause a malfunction in the reproductive abilities of the nematodes. So I've had several people work on this project before me, so I'm like 3rd generation now on this. They have previously done a lot of evolutionary conservation and analysis that way. Basically, the more conservation you have in a gene, the more important it is to the function or structure of whatever it's expressing. So here, between a human and a horse and a little worm, they all code for very, very similar things. So a mutation that changes this amino acid is likely to cause a serious issue. This has also been kind of, I guess you'd call it a bit of a simulation that you would use of a polyphen 2 prediction to input this genetic mutation and kind of see if it would cause any damaging effect on the overall gene. So it has been considered possibly damaging, which hopes this is actually the proper gene mutation site where this would actually occur. I've worked more with the PCR and gel electrophoresis side of things, so polymerase chain reactions with primer sets that have been made. Basically, this is going in and amplifying a very specific region of DNA that we want to work with, and that is visualized on a gel electrophoresis, which is right here. So these bands are just cut portions of amplified DNA. The goal is to have these as very nice, thick, singular bands that we're able to use in experiments in the future. So I have not fully gotten to what we want to call like optimized temperatures for this project yet. As you can see, I'm very close. This primer set has mainly this one band that I'm looking for. This one, however, has a few, I don't know if you can see it from here, but there's a few faint bands up here in this region and a few below. So to fix this down the road, we would want to do what we do as a gradient PCR, which is taking a bunch of temperatures, say like between 60 and 70 degrees at like 8 different temperatures, and trying to really optimize what does this primer set really work best at, so that we can get the best results of our experiment. Once that has been completed and figured out, the goal would be to use CRISPR-Cas9 to basically generate a mutation in the GON2 gene, which would be used during using CRISPR-Cas9 technology. This would be doing micro-injections to be able to test the actual function and see how this will impact this gene. So congenital stationary night blindness is associated with this variant of uncertain significance and is predicted to be damaging in the ortholog of GON2 in a C. elegans model. That is all I have. If you guys have any questions. Yes.
So if you do use CRISPR, cause the mutation on purpose, I'm guessing in nematodes?
Yes.
Would you do that over and over again to try to zero in with like what the percentage chance of damage? So will that your guess on the prediction be more accurate, or what else would you learn besides how often things go wrong?
Yeah, so the hopefully way later down the road, if this does, induce this mutation, if they are not reproducing like they're supposed to, it will give us a specific location as to, okay, this mutation of this one amino acid is what causes this. So that would contribute to possibly gene editing in the future of being able to cure this illness or this condition. So that's kind of the overall goal, is to be able to contribute to that.
And that was going to be my question. Well, the end goal, I
Yeah.
would assume, would be to reverse or to cure this.
Yes, absolutely.
Okay.
Because in my opinion, if the cells are already there and the pathway is kind of already there, and you're just giving, you're just like, hey, gene, can you please put this one channel in? Then that might be able to work.
And you may not be able to answer this precisely,
Okay.
but kind of in a target range, how far away do you think y'all are year-wise from reaching maybe a targeted gene therapy?
Probably after I would graduate.
Sure.
Yeah.
I was gonna say.
Yeah, we're hoping to be able to do micro injections and that kind of thing to really target if it is the proper location site of this mutation. By maybe the end of the year is my goal.
Okay.
So, and that is also when I would be graduating. So hopefully whoever would take it on would be able to look at that further.
Congratulations on graduating.
Thank
Yes.
you.
But thank you.
Yes.
Well done.
Other questions first?